professor at KTHM College Nasik at Department of Microbiology. Today we are going to discuss topic from MB course 363, that is metabolism, in that we are going to cover all about the transport mechanism, specifically active and passive transport. Let's begin. Content of the topics are plasma membrane composition, active transport and its types, passive transport and its types, again group translocation of bacteria, and different types of proteins used. At the end of this section, students should be able to understand how the transport of mechanism occurs across the plasma membrane. Also, difference between active and passive transport, and difference between carrier and channel proteins. To learn about transport, how exactly occurs at specific sites. Let's begin with the introduction. Plasma membrane consists of double layer of phospholipid bilayer consisting hydrophobic core facing outwards and hydrophobic tail facing inwards. Lipid bilayer consists of majorly they allow the passage for non-polar molecules as compared to polar molecules. Large molecules and iron cannot be cross the plasma membrane, so we need transport proteins to carry out the transportation of that substances. There are various types of transporter proteins used for transport mechanism. In that, mainly they facilitate the transport molecules across the hydrophobic core. So there the two proteins which are used mainly in this type of transport mechanism are channel proteins and carrier proteins. So to learn about the process, let's discuss the next point, that is the movement of substances across the membrane can be either passive, that is along the concentration gradient, and active, that is against the concentration gradient. So channel proteins and carrier proteins are used to transport in the two ways, that is in the passive transport in facilitated diffusion and in active transport. Channel and carrier proteins are the two types of proteins that are used mainly specifically in passive transport and active transport. The main difference between channel and carrier proteins are channel proteins are specific in nature, carrier proteins are not specific in nature. Both the types of proteins are transmembrane proteins spanning the multiple times. Also, channel proteins have pores, they have a channel pore inside the membrane, whereas carrier protein doesn't contain a pore. Instead, they can have a conformation sites or they can flip their conformation, and channel protein cannot flip their conformation. The rate of the channel protein is very high as compared to carrier proteins. So how these are used, let's see. There are the first type of diffusion is nothing but the passive transport. In this type of transport of molecule occurs across the membrane simply by passive diffusion, that is through a simple lipid bilayer. There are three types of passive transport. First, simple diffusion. Second, facilitated diffusion. Third, osmosis. Before that, I would like to say some of the few points for passive transport. Some non-polar molecules such as CO2, N2, oxygen, water, glycerol, ethanol can easily pass through this lipid bilayer. Why? Because the lipid bilayer allows the small transport of small molecules rather than large molecules. Also, these are limited to non-polar molecules rather than polar molecules. So, for the transportation of the polar molecules and ion channels, we need the transporter proteins. In the simple diffusion, what occurs is molecule of a substance will move from an area of a high concentration to an area of a low concentration through a lipid bilayer. All the non-polar molecules and polar small size molecules can easily pass through this type of diffusion. Mainly, example like CO2, N2, urea, methanol. So you can see in the diagram that a channel protein is present inside the plasma membrane, which helps to transport all the polar molecules and ion channels. In the facilitated diffusion, there is transport of substances occurs across a membrane from with the help of transporter proteins. and they carry out the transport of proteins such as polar large size. Again, vacillated diffusion is powered by potential energy of a concentration gradient and does not require the expenditure of metabolic energy. The rate of transport of molecule is very high as compared to simple diffusion. In this type of transport mechanism, no metabolic energy is used instead, and also the transport of molecules occurs across the along the concentration gradient. So, as you can see in the below diagram, first represents the simple diffusion which occurs naturally, that is the all these small non-polar molecules and polar molecules can easily pass through a lipid bilayer. In the second diagrams, you can see there is the transport of proteins or molecules is occurring with the help of a transporter proteins that are like channel proteins. As you can see, the third one, facilitated diffusion, is mainly for the specific type of proteins or a transporters, and the third one is nothing but the diagram for osmosis. Osmosis. It's a spontaneous net movement of solvent molecules through a selectively permeable membrane from an area of a higher water concentration to an area of lower water concentration. Higher water concentration denotes where the solvent molecule is high and solid molecules are low, and lower water concentration represent higher solute molecules rather than, and osmosis is powered by the potential energy of a concentration gradient. So, there are osmosis mainly depends on tonicity, and tonicity represents the strength of the solution. In the isotonic solution is where a relative solute concentration of two solution is equal, and in hypertonic solution the relative concentration of one solution is slightly higher as compared to other. In the hypotonic solution, where relative solute concentration are lower as compared to another solution. These are the three types of tonicity which represents in the osmosis. The molecules which move across the membrane are with the help of channel proteins. Channel proteins play a major role in this type of transport system. Molecules move across this membrane passively through an aqueous pores. So, the best example for the channel protein represent an aqua protein, aquaporins. They consist of a filter protein which helps to carry out the transporter molecules of polar small size. This let's see an overview of the passive transport.
Passive transport includes diffusion, nonspecific and specific facilitated diffusion, and osmosis. All of these processes occur along electrochemical gradients and do not require the expenditure of energy in the form of ATP. Diffusion is the net movement of a chemical down its concentration gradient, that is, from an area of high concentration to an area of lower concentration. This diffusion requires no energy expenditure by the cell, as the concentration gradient drives the diffusion. Small uncharged molecules, such as dissolved oxygen, dissolved carbon dioxide, small alcohols, and small fatty acids, can freely diffuse through the cytoplasmic membrane. Once equilibrium is reached, molecules will still move across the membrane, but there is no net movement in a particular direction.
Then discuss the second type of transport, that is active transport. Active transport is a process where a transport of molecule occurs across the membrane with the help of energy gradient, that is it occurs against the concentration gradient, that is from lower concentration of area to a higher concentration of area. The energy is provided by a proton motive force, that is by using the hydrolysis of ATP. Proton motive force is nothing but an energy gradient resulting from an hydrogen ions protons moving across from a membrane from greater ion concentration to a lesser ion concentration. Transport of molecule in this type of system is mainly carrier proteins. They carry the molecules transport against the concentration gradient. These carrier proteins bind to a large molecules like protein from one side of the membrane and release to the other side. The binding of the molecule to the carrier protein changes the conformation of the later. Means carrier proteins can, as I have said, carrier proteins can flip its conformation. How it flips? As soon as the molecule enters the carrier protein, it will attach firstly in the hydrophobic core and then it will be released in the cytosolic side. Means it changes its conformation two times. As you can see in the following diagram, represents a lipid bilayer consisting of accessory proteins also and carrier proteins. As you can see, the molecule is a molecule can able transport from one side to another with the help of using an energy, that is ATP, that is a proton motive force is generated by using the hydrolysis of ATP. As you can see, as soon as the ATP hydrolysis occurs, the energy which is released during this type of step is usually help it usually helps to carry out the transport of molecules from one side to another side. There are two types of active transports mainly. First is primary active transport and second is secondary active transport. Primary active transport is nothing but it uses a direct ATP, that is adenosine triphosphate. So it is called as a direct active transport because it uses a metabolic energy to transport the molecule across the membrane. Secondly, secondary active transport is nothing but a second type of transport that uses an electrochemical gradient instead of using a metabolic energy. An example of the primary active transport found in the many of the gram-negative bacteria are ATP binding cassette system, that is ABC system. So ABC system consists of four domains. See, before that, I would like to explain that ATP binding cassette system consists of four majorly domains, which of them two are transmembrane protein domain that are embedded in the hydrophobic core of the lipid bilayer, and the other two membranes or the domains are the cytosolic domains, which are present nearly in the facing toward the cytosolic side. So among the role of the first two, that is transmembrane domain, is to import the ATP. As soon as the molecule will move towards it, it will be located or it will be taken to the transporter domain. As soon as it comes in the transporter domain, and hydrolysis of ATP occurs in the cytosolic side of the domain that are present in the peripheral sites. So as soon as the ATP hydrolysis occur, the energy released in this type of system is used to transport the molecule against the concentration gradient. Means what? Where the molecules are especially in lower concentration, basically the proteins where they are lower in concentration, it can be transported there. As we have seen in passive transport, it occurs vice versa, means it occurs along the concentrations from higher to lower, but in active it occurs mainly from lower to higher by using the extra or coupling energy, that is ATP hydrolysis. So in the step 1, the periplasmic binding protein firstly picks up the molecule to be transported and carries it to the transmembrane domain. In the second step, the molecule to be transported across the membrane is hydrolyzed by using the ATP energy. Then energy provided by the using of ATP hydrolysis and the phosphate and the energy move across the membrane. So with the help of this type of energy, the molecule is successfully transported across the membrane. That molecule would be charged or ions. Okay. In the secondary active transport is also called as co-transport. So why it is called as a co-transport? Because there is no coupling of ATP. Instead, it is relies upon the electrochemical difference or potential difference created by the pumping of ions in and outside the cell. So co-transported can be of two types, like symporters and antiporters. Symporters carry two molecules across the membrane in the same direction, while antiporters are carry the two species or ions. Solutes are pumped in the opposite direction across the membrane. The potential energy used here is electrochemical gradient or potential difference, which is generated rather than using the metabolic energy or hydrolysis of ATP. As you can see in the diagram, first that is symptotal. Here two molecules, they may be same or they may be different. They can be transported from an area from lower concentration to an area of a higher concentration, but it occurs in the same direction. In the antiporters, you can see the transport of two molecules that may be same or different. They can be transported across the membrane with the help of antiporters in the opposite direction, that is, from an area of a lower concentration to an area of a higher concentration. The another form of active transport that is mainly used in the bacteria is a group translocation. Basically, in this type of translocation, sugar is transported across the membrane by using a phosphophenol pyruvate molecule. This process, sugar becomes phosphorylated and is transported across the membrane, like the phosphophenol group which is present in the PEP is eventually transported into the sugar via a several proteins. Here, different types of proteins are used for the translocation of the sugar in the bacteria. There are majorly four proteins. First is even protein that helps to transport the phosphoryl group, that is enzyme 1. It is also called as HPR, that is heat stable protein. It is namely as a histidine protein. Firstly, phosphate group is transported to this type of protein. Later on, from the phosphate is transferred to another type of protein, that is enzyme 2, because it has a histidine residue in its cytosolic domain. Whereas the other difference is that the phosphate group is again transferred to another group or protein called as a E2B, that the phosphate group is usually transferred because it has a cysteine in its domain. Finally, when the phosphate comes to the cysteine domain, it is attached to the sugar, and the sugar becomes final as a glucose 6 phosphate. Step 2. As soon as the glucose 6 phosphate sugar is transported across the membrane, there is no backup. Means what? Once the glucose has been converted into glucose 6 phosphate, means when it is transported across the membrane, it can no longer go back or outwards. Means the plasma membrane only allows the simple sugar to be transported. As soon as it becomes phosphorylated, plasma membrane doesn't allow because the sugar content gets charged, that is negative charge, because of the phosphophenol pyruvate, which is helping to transport the phosphate group. So this was the group translocation of sugar, which is the main active transport which occurs in the bacteria. So this is the main type of active transport. So let's see an overview of this transport.
Group translocation is a process in which an organic molecule, such as glucose, is transported into the cell while being chemically modified. The PEP phosphotransferase system is an example of group translocation. In this process, the phosphate from the high-energy compound phosphoenolpyruvate, or PEP, is transferred to HPR by enzyme 1, and the product pyruvate is released. The phosphate is then transferred from HPR to enzyme 2B by enzyme 2A. The membrane-bound enzyme 2C then carries the glucose across the membrane, and the glucose becomes phosphorylated as it enters the cell.
References for the below topics are Wikipedia, and SlideShareNet. Summary. In summary, we are going to discuss all about the starting points, like how the transport molecules occur, passive transport and its type, active transport and its type, carrier proteins and channel protein, and group translocation of bacteria. Mainly, what happens, we saw plasma membrane consists of a two phosphobiolipid layer, that is a hydrophobic core facing inwards and hydrophobic tail facing outwards. Okay, Basically, plasma membrane serve as a barrier, or it defines as a boundaries to carry out the transport molecules. So we have discussed the two types of transport system, basically passive transport and active transport. Passive transport consists of, it is the movement of molecules which occurs across the membrane, that is along the concentration gradient from an area of a higher concentration to an area of a lower concentration with the help of transporter proteins or just by the simple diffusion. And that we saw three types of transport, that is a simple diffusion, facilitated diffusion, and osmosis. In simple diffusion, we saw the movement of molecules from an area of a higher concentration to an area of lower concentration through a lipid bilayer. Here, no transport proteins are required. Example for simple diffusion are CO2, O2, ethanol, glycerol, all the steroidal hormone, etc. Also, we saw the rate of concentration of the simple diffusion is directly proportional to the concentration gradient. And we saw the second type, that is facilitated diffusion. In this type of diffusion, the movement of molecules across the membrane occurs against that is along the concentration gradient, that is from an area of higher concentration to an area of lower concentration with the help of transporter molecules. The transporter molecules which are included in this type of transport is channel proteins. Channel proteins, in that we saw the channel proteins are the proteins that are nothing but the transmembrane proteins which spans the plasma membrane multiple times. Also, we saw the examples of facilitated diffusion may be like of example of channel protein like aquaporin. Also, we saw the third type of diffusion, that is nothing but the osmosis. Osmosis is nothing but the net spontaneous movement of a molecules, that is solvent molecules, from an area of a higher water concentration to an area of a lower water concentration through a semi-permeable membrane. In this type of transport system, we saw the molecules tends to uh, go in the direction where the area of the solute is less, that is, there is the higher water concentration. And also, we saw like the tonicity depends on the osmosis. There are three types of tonicity, that is hypertonic solution and hypotonic solution and isotonic solution. Isotonic solution is a solute is where a relative solute concentration of two solutions are equal. In the hypotonic solution, relative solute concentration of one solution is slightly higher than other. In hypotonic solution, uh, a relative solute concentration of one solution is comparatively much less as compared to another solution. So, this was the three types of passive transport. In active transport, we saw there is two types of active transport. First is the primary active transport, in which we saw that it occurs with the help of using a metabolic energy, that is by using the hydrolysis of ATP. And in the active transport, primary active transport is also called as a direct active transport. In the secondary active transport, we saw it occurs usually with the help of electrochemical gradient or potential difference rather than using an ATP hydrolysis or a metabolic energy. So this is also in this type of system, and it is also called as indirect ATP hydrolysis. So the second example of primary active transport was an ABC system, that is ATP binding cassette system. In this, we saw that the transfer of molecule takes place across the membrane with the help of the transporter domains. Means there are four transporter domains: two were the transmembrane domains and two were the peripheral or cytosolic domain, which were present in the peripheral side. As soon as the molecule comes from the outside, it goes in the first transmembrane domain, and to pass it on, an ATP binds to the cytosolic domain, and hydrolysis of it occurs, and the energy released during this type of process is used to carry out the transport of molecule across the membrane. So this we saw usually which occurs in bacteria, that is EBC binding cassette system. In the secondary active transport system, we saw it is called as a co-transport system. Why it is called it as a co-transport system? Because it usually have it carry out the transport with the help of two transporter system, that is symporter and antiporter. In symporter, we saw the transport of molecule occurs in the same direction, that is they may be two different or same molecules across the membrane by using this importer. In the antiporter, we saw the transfer or molecules takes place across the membrane in the opposite direction, that is from an area. This body transfer occurs against the concentration gradient, that is from an area of a lower concentration to an area of a higher concentration. Here there is no use of metabolic energy. So this is also called as an indirect active transport system. Lastly, we saw the third type, that is the another type of active transport which occurs mainly in bacteria. This type of system is called as a group translocation of sugars, which actually occurs in bacteria. How it occurs mainly? The sugar which is transported from outside in the cell is firstly goes into the it is taken inside with the help of four different proteins, and a phosphate group is attached to that sugar so that it will be used for the further process. Firstly, enzyme E1 helps, then E2 helps, and then E2B helps. As the first one is also called as HPR, that is even protein. Why? Because it is a heat stable protein. It carries or it helps to transfer the phosphate group from a first, that is PEP, from it transferred the phosphate group from PEP to another, that is E2B, and E2B has a histidine residue in its cytosolic domain. It transferred this phosphate group to another, that is E2B protein, which has a cysteine residue in its cytosolic domain. So as soon as the phosphate group comes in this phosphate group is transferred to sugar, that is glucose, and finally glucose become glucose 6 phosphate, and this glucose 6 phosphate is used for the further process. As the best advantage of this type of transport proteins or this type of active transport is, as soon as the sugar becomes phosphorylated, it can no longer go back, or it can no longer go outside the membrane. Why? Because the plasma membrane is impermeable to charged ions, as sugar will have a charge when it will be phosphorylated. It will be having a negative charge. Just plasma membrane only allows the simple sugar rather than charged sugar. Also, we saw the different types of protein, that is a carrier and a channel protein. The difference between the carrier and the channel proteins are both the proteins are transmembrane proteins spanning, these are integral membrane transmembrane proteins which span the plasma membrane multiple times. Channel proteins are specific in nature, while carrier proteins are not specific in nature. Channel proteins have a fixed conformation, while carrier proteins can flip their conformation. The transportation rate for channel proteins is much high as compared to carrier proteins. The rate of transport rate of carrier protein is much less. And also we saw that the channel proteins have an pores in their membrane, while the carrier proteins doesn't have the pore in the membrane. The carrier channel protein cannot flip the conformation as it has a fix. As carrier proteins can flip their conformation as soon as the molecule comes inside. It flips its conformations, and so these were the following types. Sorry. First, the channel proteins are made up of lipoprotein in nature, while carrier proteins are glycoprotein in nature. So, I hope you have understood all today's topic. Thank you.